Hello again. After learning about transport in human in the previous video, today we're going to learn about transport in plants, which is subtopic 8.3. At the end of this topic, you should be able to state the pathway of water movement in root, explain the lateral pathway of water and minerals transported from surrounding soil to the root vascular system, state the water and mineral movement via xylem in stem, describe water movement via xylem by transpiration, cohesion, tension mechanism, and root pressure. State the pressure flow hypothesis in phloem, and explain the pressure flow hypothesis in phloem. Water and minerals from the soil enter the plant through the root hairs, which is part of the epidermis of roots, cross the root cortex, endodermis, and finally pass into the xylem in the vascular cylinder. Due to water potential gradient between root hair cells and soil, water from the soil diffuse into root hair cells by osmosis. There are three main lateral routes or pathway of water and minerals from surrounding soil to the xylem. First is the apoplastic route, second is the symplastic route, and the third one is transmembrane route. In apoplastic route, water and minerals are transported along the cell wall without passing through the membrane. Casparian strip, a waxy layer, blocks apoplast pathway at endodermis. Water have to enter the stele through symplastic pathway. In symplastic route, water and minerals are transported through the cytoplasm or protoplasm and then move to the neighboring cells through plasmodesmata. In transmembrane route, as the water and ions moves along apoplast, some water and minerals are transported into the protoplast of cell of epidermis and cortex and then move inward via the symplast. The figure shows the pathways of water and minerals following three routes until it reaches the xylem vessel. First is the apoplastic route, second the symplastic route, and the third one is the transmembrane route. Then they enter the endodermis tissue by symplastic route due to blockage by Casparian strip. Finally, they are transported into the vascular cylinder by symplast and apoplast routes until it reaches the cell wall of xylem and discharged by apoplast since xylem lack protoplast. In the xylem vessels, water and minerals are transported by bulk flow upward into the shoot system. The figure shows the structure of xylem. Xylem consists of vessel elements, tracheids, fibers, and parenchyma, which is not shown in the diagram. Tracheid is a tubular cell, thin, with tapered overlapping ends joined by pits that allow lateral movement of water between neighboring cells. Vessel element is a tubular cell, which is wider, shorter, thinner walled, and less tapered than tracheid. They are aligned end to end, forming long pipes known as vessels. The end walls of vessel elements have perforation plates that enable water to flow freely through the vessels. Both tracheids and vessel elements are hardened with lignin and dead at functional maturity. The hardening provides support and prevents collapse under the tension of water transport. Water and dissolved materials will move upwards as xylem sap. The movement is against the gravity. There are two mechanisms involved in the process: root pressure or pushing force and transpirational pull or pulling force. Cohesion and adhesion in the xylem vessels facilitate the transport of water by bulk flow. Root pressure is a pushing force that pushes water upwards, but only a few meters at most. Continuous pumping of mineral ions into the vascular cylinder by root cells causes low water potential in the xylem. The water potential gradient will cause water from root cells to move into the xylem by osmosis. This increases hydrostatic pressure in the xylem. This positive pressure or pushing force pushes xylem sap upwards. Transpirational pull is the force that aids in pulling the water upwards due to negative pressure potential or tension caused by transpiration. The transpirational pull on xylem sap is transmitted all the way from the leaves to the young roots and even into the soil solution. In the xylem vessels, cohesion or cohesive force between water molecules due to hydrogen bonding helps in the pulling of xylem sap from above in unbroken column or without the water molecules separating. Adhesion, the attractive force between water molecules and the hydrophilic walls of the xylem cells, helps in xylem sap rising against the gravity. Combination of tension, cohesion, and the adhesion makes water move up the stem. The negative pressure potential that causes water to move up through the xylem develops at the surface of mesophyll cell walls in the leaf. The cell wall acts like a very thin capillary network. Water adheres to the cellulose microfibrils and other hydrophilic components of the cell wall. The figure shows how transpirational pull is generated. Negative pressure or tension at the air-water interface in the leaf is the basis of transpirational pull, which draws water out of the xylem. First step, in transpiration, water vapor diffuses from the moist air spaces of the leaf to the drier air outside via stomata. In second step, at first, water vapor lost by transpiration is replaced by evaporation from the water phloem that coats mesophyll cells. In the third step, the evaporation of the water phloem causes the air-water interface to retreat farther into the cell wall and to become more curved. The curvature increases the surface tension and the rate of transpiration. In the fourth step, the increased surface tension shown in step 3 pulls water from surrounding cells and air spaces. In the final step, water from the xylem is pulled into the surrounding cells and air spaces to replace the water that was lost. This figure shows the ascent of xylem sap and how transpirational pull on xylem sap is transmitted all the way from the leaves to the roots. Hydrogen bonding forms an unbroken chain of water molecules extending from leaves to the soil. The force driving the ascent of xylem sap is a gradient of water potential. For bulk flow over long distance, the water potential gradient is due mainly to a gradient of the pressure potential. Transpiration results in the pressure potential at the leaf end of the xylem being lower than the pressure potential at the root end. The value of water potential shown may vary during daylight, but the direction of the water potential gradient remains the same. The next video shows the transport of water in plants.
In vascular plants, water and dissolved inorganic nutrients from the soil are transported upward against gravity in a cylindrical network of tubes called xylem that runs all the way from the highest leaves to the tiniest tips of the deepest roots. The process begins when diffusion and osmosis draw water into millions of microscopic root hairs and into the xylem of the roots. This event results in a positive pressure called root pressure that actually pushes the sap a short distance up the xylem towards the leaves. Then the forces of adhesion take over as the water molecules, which possess weak electrical charges, cling to the walls of the xylem tubes. At the same time, cohesion between the electrically charged water molecules causes a long, unbroken column of water to form inside the xylem. In fact, it is these same forces that cause perfect spheres to form when water droplets splash. Cohesive forces also create the surface tension of water, which produces the strong elastic film seen here. Working together, the forces of cohesion and adhesion produce an effect known as capillary action that causes liquids in narrow spaces to flow upward. However, none of these activities are as important to upward water movement as a process called transpiration that takes place inside the leaves. During transpiration, evaporation causes water to escape into the atmosphere through tiny pores in the leaves called stomata. As water is pulled from between the microfibrils of the mesophyll cell walls, crescent-shaped menisci are formed. As these become more curved, the surface tension increases, creating a negative pressure that pulls water out of the cells and eventually out of the xylem, resulting in what is called transpirational pull.
Vascular plants produce nutrients such as sucrose in their leaves by photosynthesis. These nutrients must be transported to the rest of the shoot or the root tips where growth occurs. The nutrients are transported in a process called translocation. Translocation is the movement of organic solute or phloem sap from the leaves or source to the sieve tubes and to be carried to other parts of the plant or sink. Phloem sap moves through the sieve tubes by bulk flow driven by positive pressure, known as pressure flow. The pressure flow hypothesis explains why phloem sap flows from the source to the sink. Sugar source is the sugar production organ in plant where photosynthesis occurs. Soluble organic products are synthesized. Examples of sugar source are green leaves. Sugar sink is the region in plant where soluble organic products is unloaded from sieve tube to be utilized or stored. Examples of sugar sinks are growing roots and shoots, stem, fruits, and flower buds. The figure shows the bulk flow by positive pressure or pressure flow in a sieve tube, the building of pressure at the source, and the reduction of that pressure at the sink cause sap to flow from source to sink. First step, loading of sugar in the form of sucrose into the sieve tube at the source, for example a mesophyll cell in a leaf, reduces water potential inside the sieve tube elements. This causes the tube to take up water by osmosis. Second step, this uptake of water generates a positive pressure that forces the sap to flow along the tube. Third step, the pressure is relieved by the unloading of sugar mainly by passive transport and the consequent loss of water at the sink. And finally, in leaf-to-root translocation, xylem recycles water from sink to source. Sinks usually receives sugar from the nearest sugar sources. With this, we have finished learning about transport in plants.